Recording in progress.
All right, that is ready to go. All right, today's lesson, we're going to focus on this protein secretory pathway. So like I said, we've learned everything about gene expression, how genes act as the instructions to make proteins. So we've covered all the way up to a polypeptide chain, right? That's the end of translation. You end up with a polypeptide chain. So now we're going to look at what's the next step after that. Because between a polypeptide chain to a functional protein, there are still steps that need to be taken, right? So today's lesson is going to focus on that. So you're going to identify and explain the roles of organelles in creating a functional protein, and then also to be able to define what exocytosis means. Okay, so remember the organelles that I keep showing you guys with the model of a cell. Each one of those organelles plays a role in the maintenance of a cell. But these organelles, like rough endoplasmic reticulum, Golgi apparatus, and the vesicles associated with it, are the key organelles that need to be functional for a protein to be made. Okay, so under your study design, you guys are expected to know the role of your rough endoplasmic reticulum, your Golgi apparatus, the associated vesicles, and how they work together to get a protein molecule from the ribosome as a polypeptide chain all the way to the outside of the cell where it will actually serve a purpose. All right, before we get started, this model is showing with or without tryptophan. First question, can I get Janita? Do you see any tryptophan here? Yeah, actually, I copied these questions from Ms. Long's side, so this one's kind of confusing to me. Disregard that. Okay, number 2. RNA polymerase binds to what site? Can I get Nani? Where does RNA polymerase bind to? Promoter, okay? Always ask yourself, what does the RNA polymerase do? It creates a mRNA copy of the gene. And for it to do that, it needs to start at the promoter and then go from there. Okay? Regulatory genes produce. Oh, you're supposed to give the answer. All right, can I get Abi? What are the 2 regulatory proteins that regulatory genes can make? There's only 2 examples that we were given. So these are the 2 regulatory proteins that can be made using regulatory genes. I'll give you a hint, one of them is already there. So this is talking about remember how in an operon, so this is the operon, that's all about making the tryptophan or lactose. But I'm talking about the regulatory gene and the protein that makes. What is the proteins that it can make? There's 2 proteins. One is up here, if you can spot it. The other one is the one that only eukaryotes usually deal with. Their name very much explains what they do. Who said that? Yeah, Mayor? Yeah, simple as that. Activator and repressor proteins. Okay, they literally state what they do, right? Repressor proteins act to repress gene expression. Activator proteins act to basically activate gene expression. So repressors are the ones you guys need to focus mostly on because they work with prokaryotes, and under your study design, you guys really need to focus on prokaryotes for gene regulation. Eukaryotes for gene regulation are not covered for you guys as much. All you need to know is that activators work for eukaryotes, and that should be enough already. All right, so let's see. Can I get where does a repressor protein bind to? Sorry, okay, let me make this clear. The regulatory promoter and the gene, it is what makes the protein. Okay, this part needs to exist for a repressor protein to be made. But where does the repressor protein bind to? The operator. Okay, so they can only function, the regulatory proteins can only function when they bind to somewhere on the operon, and specifically the operator, because that is the binding site for regulatory for repressors specifically. Okay, so would happen? Special gene, what would happen to the image? All right, let's see. Gavin, after transcription of the structural genes, what would happen to the mRNA? So this is a little bit of a outside the box question because we didn't really cover this. But what do you think would happen to the mRNA after transcription has occurred? So transcription of the structural gene, what would happen to the mRNA? Translation, yeah. Okay, but here's the question. What do you think happens to the mRNA after translation? So basically, it served its function. What happens to the mRNA? Any ideas? What do you think would happen? What's the next logical step? It's going to be degraded, and then basically its parts are going to be recycled to make more mRNA copies later on. Because they have to use the same parts over and over again. So that's why they're recycled and then reused. Okay, this one's a bit of a, it sounds complicated, but it's basically reading comprehension. Try to understand what it's saying and answer the question. Done. Are you looking at the slides? I trust you, year 12, so I don't need to monitor your slides. All right, another 30 seconds. All right, can I get, sir? Guess. Don't guess, try to answer. Like I said, guys, it sounds complicated. In biology, you're going to face these kind of questions that sound like you've never learned it before, right? You've never learned about lens cells or crystalline or liver cells. You didn't know about that. What they're trying to get you to think about is what you've learned about basically translation, transcription, regulatory proteins, all this related into this question, okay? So let's break this down. Activated molecules that are present in the cell can turn genes on, right? That is a regulatory protein. So activators will turn the gene on. Only liver cells produce that protein, and only lens cells produce crystalline. The difference in protein production between these two cells can be explained by, so we go through each answer and see whether it actually makes sense in the sense of what we know about gene expression, about regulatory proteins. Based on that, we can cut out the answers, right? So let's go with both cells containing identical activated proteins, molecules. Remember, each regulatory protein is unique to each operon and each gene it controls because it needs to bind to that operator. So it needs to be specific to that operator. Okay, each operator is going to be different. Each gene is going to be different. So they're going to be very specialized. Not all activated proteins can work for all molecules. They need to be specific to each function. Okay, liver cells not having the gene that codes for crystalline. Okay, so so the presence of RNA polymerase in lens cells, but not in liver cells. So that's not right because it can code for the protein, so that's not accurate. Liver cells not having the gene that codes for crystalline. That's like this. The answer is A. Because the best thing to note, right, rather than saying liver cells do not contain the gene for crystalline, why is this wrong? Who can tell me? It's actually a very straightforward answer of why C is wrong. What is the answer stating? Liver cells not having the gene to code for that protein. Remember that where do genes come from? DNA, right? And is DNA the same for every cell in the body? Except for your reproductive cells, it's all the same. Like every other cell in your body, except for your reproductive organs, have the same genetic code. It's the same DNA, basically the same textbook that they derive information from. So it's going to have that gene. it's going to have all the genes in your body is going to be present in your liver cells as well. But the difference is it's not making or expressing that gene. Whereas with A, it's specifically talking about the activator molecules and how it is for the gene coding for crystalline. So we know that the activator molecules are specific to the crystalline, and we know that crystalline is only made at the lens cells. So that is the appropriate answer. Okay. They want you to remember everything you've learned and reapply it. Basically, they want you to know that RNA polymerase is going to be present as long as there's protein being made, because that's the first step, right? It needs to be transcribed. Second, they need you to know that DNA is the same for all the cells, regardless of where it's located. So it will have all the genes for every part of your body. It's just that your body, your cells are so specific of which genes they need to express. So they know where to find those genes in your DNA and then express them. And then the last one, they want you to remember that regulatory proteins are specific to the gene that they are regulating. And they are going to be specific in terms of their shape and also their function. So they are definitely not identical when it comes to the regulatory proteins. Anyone still confused about this? All right, let's actually get into the lesson for today. Today's lesson is not going to be very intense because it's just going to talk about a few steps and then we're going to do more revision. All right, so basically, we know so far that cells act as protein factories, right? There are ribosomes in the cells, and those ribosomes make proteins, and those proteins are sent throughout your body for different reasons, right? So we know for a fact that cells make those proteins, but one thing to note is that not all the proteins made by cells are used by those cells themselves, okay? Those cells usually release those proteins and they will look for their specific target cell. So think about it in terms of an example such as, where is insulin produced in your body? Can anyone tell me? Insulin. No? Okay. It's produced in your liver cells, if you don't know. But the fact is, insulin is going to be functioning in your blood. Okay, that's where its primary function is going to be. So insulin needs to get from your liver into your bloodstream for it to actually have an effect. It's the same thing with a lot of proteins made in your cells. They need to leave the cell and get to that targeted cell to actually have a function. Okay, so this is the same for hormones, enzymes, toxins, and all kinds of antimicrobial peptides. So another example, a lot of the hormones that you guys are using in your body is actually made inside your brain. A part of your brain makes a lot of those hormones, but the fact is their function is going to be all over your body. Those hormones need to travel in your blood and then need to get to that specific target cell, be released, bind to that target cell, and then only they will have an effect, right? So another example is that proteins such as hormones can actually be transported in the bloodstream to get to where they need to go. Not all proteins travel through the bloodstream though, but that's a good example to use if they ever ask you about it. Just remember, proteins made in the cell are not always used in the cell themselves. So far what we know about protein synthesis, we've talked about the starting step of transcription, moving on to RNA processing, then moving on to translation, and then now we're at the step where we have a polypeptide chain ready to go. The next step is to get it to fold, okay? No point having a polypeptide chain because that is not functional. It's not going to carry a function. It is just a chain of amino acids. Only when that is folded into a molecule, a complex molecule, can it actually serve a function, right? It can either do transportation, can add structural integrity. It has 7 general functions it can do, right? Proteins. For it to be able to do any one of those 7 functions, it needs to experience folding. So where does it experience that? The first place is the first site that the polypeptide chain goes to is called the rough endoplasmic reticulum. But before I start talking about parts of the cell that you guys probably have forgotten from year 8 science, let me just do a quick refresher of a cell, just so that you guys are aware of what it's what and where it is. Okay, so we know that this is the nucleus, and that's going to be inside a nuclear envelope. You guys don't need to know the nuclear envelope. All you need to know is the nucleus. That is where your DNA is stored. That is where transcription happens and RNA processing happens. Only once RNA processing is done and we have a mature mRNA molecule, it is then sent outside where it will bind to one of those dots that you see. Now, who can tell me what that dot is? 10. What is that dot? Where does your where does your mature mRNA molecule go to? What's the next step? Okay, we have RNA processing. What's the next step after that? Yes, translation. So where does that happen? Yes. Yes, the answer is ribosomes. Okay, ribosome in the cell is the site of protein synthesis, and they're usually bound to this structure called the rough endoplasmic reticulum. So that's an idea you can link together right now. The fact that ribosomes are usually attached to the rough endoplasmic reticulum. They can also be found free floating in the cytoplasm, but a large concentration will be bound onto the rough endoplasmic reticulum. It actually makes sense considering what comes next, because if in your ribosome a functional polypeptide chain is made, it needs to be folded to become functional. So it only makes sense that it's connected right to the place where that will happen, right? So it will first go to the endoplasmic reticulum. So the function of the rough endoplasmic reticulum is basically to fold and transport the protein, right? So you can see it's almost like a canyon where it follows a pathway. While following that pathway, it is folded based on its characteristics and its function, and then it will follow this path until it can get to the next step, which is the Golgi apparatus. But because it's in the cytoplasm, which is the space inside a cell, so this cytoplasm isn't conducive for a protein or a polypeptide to move around. Okay, it doesn't move around very well in a cytoplasm, so it needs to be specially transported in something called a vesicle. Okay, there are two vesicles that you guys need to know, and they usually are called one or the other based on what step of the flow it's at. Okay, if it's being moved from the rough endoplasmic reticulum to the Golgi, it's called a transport vesicle. If it's being moved outside of the cell already, it's called a secretory vesicle. All you need to know is that vesicles are basically a transportation method for proteins, for molecules to get to one spot or the other inside a cell. If that is because the cytoplasm is not a good place for proteins to move around in. It's not a conducive way for proteins to move around, so they get their own special transportation method. So we'll talk about what vesicles are and how they look in a bit, but first, make sure you guys remember rough endoplasmic reticulum is the first place that a polypeptide chain goes right after being translated, and it will experience folding and it will be transported to the next part. So this is where a simple polypeptide chain will then be folded into the secondary, tertiary, and then quaternary structure as well. So always remember that folding happens here, and this is where they gain function. So next is your transport vesicle. So from your rough endoplasmic reticulum, it needs to get to the Golgi apparatus. For it to be able to transport itself in the cytoplasm, it will be covered in a transport vesicle. So a transport vesicle is actually made up of the same thing that your plasma membrane is made of. So how it works is it's almost like it peels off the plasma membrane and forms a circle around itself. So let me try to zoom in and show you guys how that looks like. All right, so when the proteins are getting to the surface of the plasma membrane, a part of it actually pinches off and forms a circle. And then that is what becomes the transport vesicle. So the transport vesicle, the layer it's made of is actually the same layer that your plasma membrane is made of, which is a hydrophilic head and a hydrophobic tail, and that forms a bilayer. So this is exactly the same configuration as your plasma membrane. That is why they can do this step where once they get to where they need to go, they will tether onto the plasma membrane of the site where they need to go, and it will actually diffuse on into itself as well, almost like it formed and then diffused where it needs to go, because they're both made using the same thing. The surface of organelles are covered in plasma membrane, and through that plasma membrane, a vesicle can emerge and diffuse back in. Anyone's confused about this? Sounds very hard to believe or see, imagine. If I'm trying to think of a good analogy for this, I'll think of one over time. Let's just keep going then. All right. So once they've become functional, they need to be transported to the Golgi apparatus. They will travel through the vesicles. These vesicles form and fuse with the membrane of organelles. And this is only possible due to the fluid nature of plasma membrane. Okay, you can't just do this with any object, right? It needs to have a fluid-like nature, and that's why plasma membranes are able to do this. So after the transport vesicle, it moves to the Golgi apparatus. That is the where proteins are modified and where the product is modified and packaged into functional proteins. This is where proteins would have had sugar groups either added onto them or removed. This could be for functional purposes. And then the last part is when it's packaged into a secretory vesicle for export. So this is the final step already, where it's put into a vesicle again, which is the same thing as we talked about just now: hydrophilic head, hydrophobic tail. It will form a circular structure, and then it will export the protein out of the cell. So a vesicle buds off the Golgi apparatus, travels through the cytoplasm, and then finally it fuses with the plasma membrane of the cell, where it will be released out. So this process is called exocytosis, right? So exocytosis, basically when proteins contained within the cell are released into the environment. It's a very simple process, but this is what allows us to get proteins that are made inside cells to get to where they need to in the body. Right? So talking about exocytosis a bit more. So when a vesicle containing a secretory product, so secretory products is a general name referring to what is being transported, the protein. But since it's not functional yet, we just call it a secretory product until it's until it's released from the cell. We will generally refer to it as secretory products. So a vesicle containing the products will be transported to the plasma membrane. The membrane of the vesicle and the membrane of the plasma membrane will fuse together and dissolve into each other. Once it does that, it will release all the products out. So you can see that in this picture here, right? The fact that it actually diffuses with the plasma membrane, the vesicle, and you can see that the structure of the vesicle is identical to the structure of the plasma membrane. That's why they can merge and fuse together as easily as they do. And it's also due to the nature of the plasma membrane, the fluid-like nature. You have to remember that, because if they do ask why can vesicles form and fuse so easily, you need to be able to refer to the fluid-like nature, the plasma membrane. So yeah, easy put, exocytosis, process by which contents of a vesicle are released from a cell. This is necessary because a large protein will require energy to be transported out of the cell. That is referred to as active transport. Okay, anytime something is moved out of the cell, it is a form of transportation. Whether it takes energy or not will be what determines whether it's active transport or passive transport. This process of getting a protein outside of the cell is incredibly energy demanding because proteins are large molecules and they need to be transported in special vesicles so that they can actually get outside of the cell. So that requires a lot of energy. Right? Any questions? All right. So if I was endoplasmic reticulum, how would you like me? Smooth, rough? Yes. Can't even. Nucleus to every organ. So this is because nucleus is basically the brains of a cell. Okay, it does the everything from giving instructions to actually making the instructions to make other things. So that's why that's important note. This one over here, surprisingly, this was actually generated by ChatGPT. So I was curious to see whether ChatGPT could generate memes. Not a great one, but it actually has some content in it. So when you think about it, when it's in the ER, it's a polypeptide chain, right? That's why there's so much of it. It's like small molecules that are connected together like that. When it's done coming out of the Golgi apparatus, it is basically going to be one complete molecule, a big protein that is folded upon itself, ready to work. So yeah, horrible memes as always. All right, that's it for today's lesson.